In this video, we're going to look at the control of oxidative metabolism. So to look at the rate of oxidative phosphorylation, we really need to look at the energetics of oxidative phosphorylation. So if we just focus on the pathway of NADH to cytochrome C, that is going to run in near equilibrium. And here's our equation. And you see we have 1/2 NADH and 1/2 NAD plus. That's because we're just giving one electron away at a time. So in effect, it's 1/2 NADH. Overall, this reaction has a delta G near 0, which means it's at equilibrium. If we look at KEQ, KEQ is dependent on the ratios of NAD plus to NADH, cytochrome and its different oxidation states, and ATP to ADP, which means, since this is near equilibrium, if we add a lot of ATP or our levels of ATP is high, oxidative phosphorylation will actually run in reverse. We'll take electrons from cytochrome C, give it to NAD+, use ATP, and make NADH. Now, the last step of oxidative phosphorylation, cytochrome C, is reversible, and this could be thought as one of our control points. And this reaction is controlled by the amount of substrate you have, the amount of reduced cytochrome C. And the reduced amount of cytochrome C, if we go back to our previous equation, depends on the ratios of NADH to NAD plus and ATP to ADP. What this means is that the more NADH we have and the less amount of ATP we have, the faster oxidative phosphorylation will be, because we will have more reduced cytochrome C, which will do the last step of oxidative phosphorylation. And this actually makes a lot of sense. NADH is feeding electrons to oxidative phosphorylation, and we want to make ATP. So if we have a lot of NADH and a lot of ADP, we want to run oxidative phosphorylation to make ATP. Now, we do have regulation proteins for ATP synthase, and that protein is called IF1. Now, if you're respiring and you have a high pH, IF1 will be an inactive tetramer. But if your pH changes and falls below 6.5, then the protein will split into dimers and go and inhibit ATPase. Specifically, you're binding the alpha DP and beta DP subunits. And this is great for us because let's say we have a lack of oxygen. We don't want ATP hydrolysis to run backwards, which it would. So ATP synthase can actually run in reverse, where you use ATP hydrolysis to run this machine backwards, wasting your ATP. You don't want that to happen if you're not doing ATP synthesis, so you just block this mechanism by more or less putting a bar into it. That's kind of what IF1 looks like. It looks like a long bar, and you just stick it in your ATP synthase, and it's not going to be running backwards. Now, thinking about control on the grand scheme, remember we get electrons from glycolysis in the citric acid cycle. These two pathways are controlled based on how much oxidative phosphorylation we're doing. And some of the enzymes you're looking for control are PFK, pyruvate dehydrogenase, citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. Now, we talked about this already, but PFK is inhibited by citrate. Now, when your ATP demands decreases, that means you have plenty of ATP and not a lot of ADP around, which means isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase both slow down, which means your citrate concentrations will increase because you're more or less stuck here at this part of the citric acid cycle. So your citrate will keep increasing. Now, citrate can leave the mitochondria by a specific transport system, and when it does, it will go to the cytosol and block PFK, which prevents any more material going to the citric acid cycle since we already have plenty of energy. Now, we've talked about how aerobic respiration makes a lot of energy, 32 ATP versus 2 ATP, but there are drawbacks for using oxygen. One, if you don't have oxygen in your aerobic organism, you will damage yourself if you go without oxygen too long. For example, your brain, you'll become brain dead if you don't have enough oxygen, or you'll eventually die. So since we are used to this much energy, we need that much energy or we will die very quickly. Also, during this, you will produce reactive oxygen species that is oxygen with a free radical, superoxide radicals. These will go and damage different cellular components. You can also take electrons from other molecules on accident, changing them to free radicals and starting this big old chain reaction. So your lipids can be affected, your DNA can be affected, enzymes can be affected, all by these free radicals. And if we look at some of the diseases ravaging us today, Parkinson's, Alzheimer's, Huntington's disease are all thought to be associated with different oxidative damage from the mitochondria due to using oxygen. And there's also a theory of aging, that these free radicals, as they build up, that's why we age, because you get more and more damage due to these free radicals. Now, there are ways to help and prevent these free radicals, and those are what we call antioxidants. They will destroy your different free radicals. You have actually a protein in your body called superoxide dismutase, or SOD, in all cells, which will take this oxygen-free radical and change it to hydrogen peroxide. And this reaction is so fast that we call it diffusion control limited. That is, the slowest step of this reaction is just the oxide coming to the enzyme. And one of the reasons why this is so fast is because you actually have electricity helping guide your free radical to the active site. So you guide this to the active site, and on the bottom you have a copper and an arginine. And you have a bunch of hydrogen bonds of glutamic acid, asparagine, lysine, and threonine at the entrance that are just guiding it to our copper and our arginine right here. Now, once you make hydrogen peroxide, you have another enzyme called catalase that breaks that down into water and oxygen. You also have glutathione, which will take this hydrogen peroxide, break it down into water, and it will form a disulfide bonds. Other oxidants we can get from plants, such as vitamin C and vitamin E, although we aren't sure these actually work in humans as antioxidants. We know there are antioxidants in plants, and so it's logical to think that, well, they'll do the same thing in our body, yet there's actually not strong scientific evidence to actually show that. So who knows if eating antioxidants like that are actually providing a big benefit. And that's it for this video. I will see you in the next one.